Assalamualaikum warahmatullahi wabarakatuh. We, Masyitah binti Zaihan, Nur Dini binti Mazlan, Nur Farina binti Muhamad Rizal, and Tasha Xuan will be presenting on a normal biological process involving G protein coupled receptor and receptor enzyme for our STBK 3322 biosignaling class. First and foremost, it is essential to know and understand the 6 genotypes of signal transducers, which are the G protein coupled receptor, the gated ion channel, the receptor enzyme, the steroid receptor, the adhesion receptor, and the receptor with no intrinsic enzyme activity. Today, we will be discussing on the incorporation of 2 types of cell surface receptors, which are the G protein coupled receptor and the receptor enzyme in a normal biological process. The incorporation of GPCR and receptor enzyme can be depicted through transactivation of the RTK by GPCR. This process generally can occur through binding of ligand or agonist to specific binding sites of G protein on GPCR, which will dissociate the heterotrimeric G protein. This will then generate second messengers such as calcium ion. GPCR-mediated RTK transactivation is dependent on matrix metalloproteinase MMPs and the A-disintegrin and metalloproteinase ADAMs. Shedding of ligand by MMPs and ADAMs allow the dimerization of RTK. This activation of RTK by ligand will cause cellular response. We unanimously chose epidermal growth factor signaling transduction process in wound healing of corneal epithelial cells as our topic.
In this presentation, we will go through what happens during the process in wound healing of corneal epithelial cells. Epithelial wound healing is at least in part of mediated in an autocrine fashion by epidermal growth factor receptor EGFR ligand interaction. We will identify the endogenous EGFR ligand and the mechanism in cellular response PRTK pathway by which it is generated in activation of receptor tyrosine kinase EGFR by HBEGF ligand binding in response of wounding human corneal epithelial cells. There are 3 main importance in the process of transactivation of EGFR by GPCR. First and foremost, it promotes all 4 processes, which are proliferation, migration, survival, and differentiation involved in corneal epithelial wound healing. Secondly, it maintains corneal homeostasis and preserving its function under pathogenic conditions. Thirdly, it plays an important role in re-epithelialization and dermal maturation in wound healing. It is a key regulator for maintaining a healthy cornea and promoting regrowth of the wounded cornea. Next, I will go on with the important components involved in this process. The ligand used in this process is lysophosphatidic acid LPA. The G protein coupled receptor used is lysophosphatidic acid receptors LPA1. Calcium ion will be the second messenger. There are 2 membrane-bound proteases, which are MPPs and ADAMs mentioned in the earlier slide. Receptor tyrosine kinase used in this process will be epidermal growth factor receptor EGFR, and lastly, RTK ligands will be heparin binding epidermal growth factor. The overview of epidermal growth factor signaling transduction. There are 3 main processes. Firstly, ligand LPA will bind to GPCR LPA1. Secondly, transactivation of ADAMs and MPPs precursors will then occur. Thirdly, activation of EGFR will then proceed.
The process of binding of ligand LPA to GPCR LPA1 firstly starts with LPA acting as a signaling molecule and binds to the G-coupled protein receptor. This causes the G protein alpha subunit to exchange GDP to GTP. The alpha subunit then dissociates from the beta and gamma subunit. This will trigger a cellular response. GTP is hydrolyzed to GDP and LPA comes off the receptor. Once that happens, the alpha subunit comes back together with the receptor and the beta and gamma subunit. GPCR stimulation triggers the generation of several second messengers such as Ca2+ ions.
Ligand LPA binds to G protein coupled receptor which located in membrane. Heterotrimeric G protein composed of 3 protein subunit alpha, beta, gamma. Alpha subunit has GDP bound dissociate to GTP. GTP then binds, cause conformational change. After the activation of receptor, LPA ligand comes off G protein receptor.
Next, MPP and atom transactivation precursors. As mentioned before, the stimulation of GPCR triggers the generation of Ca2+ ions. Ca2+ ion then trigger the activation of MMPs and atoms. MMPs and atoms are molecules that are able to cleave EGF ligand, which in this case is the Hb EGF. This promotes ligand shedding into the extracellular space. Once the ligand is shedded into the extracellular space, it is then able to bind to the EGFR and other EGFR family members. This will promote dimerization and activation of the EGFR. This is called the triple membrane passing signal. Following the binding of Hb-EGF ligands on the EGFR is the activation of EGFR by EGF. On occupation by Hb-EGF, the EGFR forms a dimer through dimerization. This induces a change in the conformation of the cytoplasmic domain that reveals its latent tyrosine protein kinase activity. Through autophosphorylation, this phosphorylates the tyrosine residues on the linked receptor molecules. The dimerized phosphorylated molecule constitutes the catalytically active receptor.
EGFR release on wounding act as a ligand that binds to EGFR monomer, promotes dimerization.
Phosphorylation of EGFR creates docking sites for adapter proteins that contain special type of binding domain, such as the PI3K domain. A variety of proteins contain this domain, and when one of these proteins bind, it can initiate a downstream signaling cascade, such as the AKT or PI3K pathway, that leads to a cellular response in the corneal epithelial cells.
PI3K signaling is initiated by Hb-EGF binds to EGFR, leading to EGFR dimerization. As a consequence, lipid kinase PI3K is recruited to the internal docking site. Activated PI3K converts membrane lipid PIP2 to active PIP3 form, which activate K signaling AKT. AKT promotes cell growth through protein synthesis driven by mTOR. In a nutshell, to wrap all these things up, LPA stimulates corneal epithelial wound healing by facilitating Hb-EGF release and providing ligand to EGFR. The phosphorylated EGFR then activates downstream signaling pathways, which leads to cell proliferation and wound closure. The activation is sufficient for PI3K pathways.